Welcome, everyone. Great to see you all here checking in for our special grand rounds. I've been looking forward to this grand rounds, my goodness, for at least a couple of years. Having known Michelle and known of her work and known that and having seen some of her other talks, just knowing what a spectacular treat we are in for. I'm going to turn it over to her PhD mentor, Dr. Alfonso Araque, who's going to tell us a little bit about Michelle and her specific work. But I just want to let the audience know that Michelle has been involved in various themes and projects in our department while she's been a student here. And we had hoped to be able to persuade her to choose a psychiatry residency, but she's doing the next best thing, which is a neurology residency. And as some of you may have heard when we were chatting together, we both agreed that these two fields are going to be coming closer and closer together. And I believe that Michelle may end up being one of the pioneers who helps bring around some of that integration. So thank you, Michelle, for being here for this special grand rounds. Thank you, Alfonso, for being the person who will introduce her and who will moderate this grand rounds. And I'm going to turn it over to you.
Thank you, Sophia. Good morning, everybody. Thank you for being here. It is my real pleasure to introduce Dr. Michelle Corkrum in this talk. I was fortunate to be the PhD advisor of Michelle. It was a real pleasure to have her in the lab. You will see she has later today that she has done a absolutely great work working in the lab. It's not in the past. We're still working together, so it's very exciting that I'm very happy to be continuing working with her. The work of the PhD work of Michelle to get her PhD was focused on the role of astrocytes in dopamine signaling in the brain, specifically in the mesolimbic system. She made a wonderful work to demonstrate that astrocytes are involved in dopaminergic signaling. This is a very seminal, important, groundbreaking work that she was able to produce. She also produced another piece of work related to the role of astrocytes in opioid signaling. So she was able to produce several groundbreaking results to better understand how the role of astrocytes in brain function, specifically in this mesolimbic system. Michelle received multiple awards during his career. One of them, are probably the most important to say, she received the NIDA F30 grant to develop her research. She received also some other awards. And Michelle is a great scientist. She's also a great physician. Unfortunately, she's going to be both physician and scientist. So I'm sure her patients will benefit from her. But fortunately, I'm glad to say that the scientific community will have the opportunity to also benefit from her contributions. She is very talented, a great scientist. I don't know as a patient, maybe you will have to treat me someday. Most definitely it's gonna be like that. We all ended up with that. But as a great scientist, you're a great scientist, Michelle. So I'm very, very happy for the scientific community that you stay in the scientific community. And without further introduction, I want to pass the word to you, Michelle, to give us your lecture. Thank you.
Thank you so much, Alfonso. And thank you so much, Dr. Vinogrado, for inviting me to give this talk today. I'm so excited to present this work. Alfonso, it was so great working in your lab, and I'm glad that we're still working together now. So I will start sharing my screen. Perfect. All righty. So today, thank you everyone for joining. I'm very excited to present my work that I conducted in the lab of Dr. Alfonso Araque, investigating the role of astrocytes in mesolimbic dopamine signaling, and also propose novel cellular targets for psychiatric disorders, focusing on the astrocyte. So I do not have any disclosures. And today I will talk about the role of dopamine in brain physiology and pathology, and then also introduce astrocytes as key cellular components of brain information processing in the reward system, and also demonstrate the role of astrocytes in the psychostimulant effects of amphetamine as well. So dopamine signaling is a very important component of our everyday activities in the world. It's important for movement, it's important for learning and memory, and it's also important for reward. And when dopamine signaling is disrupted, it can contribute to multiple psychiatric disorders, including mania, depression, dementia, schizophrenia, and addiction. So it's very important for us to be able to understand what is contributing to dopamine signaling. And to do this, I am showing a schematic of a sagittal section of a human brain, and dopamine projects throughout the brain. It originates in the ventral tegmental area and the substantia nigra, and it projects to the hippocampus, the nucleus accumbens, the frontal cortex, and also the striatum. And these multiple projections throughout the brain of dopamine is what's very important for both the physiologic and pathologic conditions associated with dopamine signaling. My project was specifically looking at the role of dopamine and reward signaling. So I was looking at dopamine signaling from the ventral tegmental area to the nucleus accumbens. And a lot is known about the role of neurons in dopamine signaling. When we think about the human brain and the cells in the human brain, we think about neurons. And a lot of research has been conducted to investigate how neurons contribute to dopamine signaling. But another major cell type in the brain are astrocytes, and not much is known about the role astrocytes play in dopamine signaling. And this is mainly because astrocytes have traditionally been considered support cells in the brain. They're not electrically excitable, they do not generate action potentials, and so they are thought to only play support roles in the brain, including providing structural support to neurons, aiding in neuronal guidance, aiding in synaptogenesis, helping with extracellular homeostasis, and then also providing trophic and metabolic support to neurons as well. But even in the 1800s, Cajal, one of the founding fathers of modern neuroscience, recognized that astrocytes are a major component of brain function. He illustrated the astrocyte front and center with a cell body and processes extending from the astrocyte. And some of these processes go to blood vessels, and some of the processes also encompass neuronal synapses, suggesting that astrocytes may also play a major role in synaptic transmission and plasticity. This was further illustrated in work that has been conducted over the last few decades, showing that although astrocytes are not electrically excitable, they do not generate action potentials. In response to brain activity, they do have this increase in calcium. And this increase in calcium can then lead to the release of neuroactive substances like ATP, GABA, and glutamate to modulate synaptic transmission and plasticity. So astrocytes do indeed play an active role in brain function. So overall, the goals of the project that I'm presenting today was to investigate whether or not astrocytes play an active role in dopamine neuromodulation. Do they respond to dopamine with increases in calcium and contribute to dopamine-evoked synaptic transmission changes? And then also the aims of this project was to investigate the role of astrocytes in amphetamine neuromodulation as well. To do this, I will first show my work looking at whether or not astrocytes respond to dopamine. So for these experiments, I did use a preclinical model. Right now, it is really hard for us to be able to investigate astrocyte calcium signaling in the human brain. So I did transition to a mouse preclinical model. And here I'm showing a sagittal section of a mouse brain where we targeted a light activated channel to dopamine neurons in the ventral tegmental area. And we were able to allow this light activated channel to travel down axons to the nucleus accumbens to stimulate dopamine transmission in the nucleus accumbens. And we were able to verify our targeting of our light activated channel by co-localizing our tyrosine hydroxylase, which is a marker for dopamine neurons, with TD tomato, which was on our viral vector. And so we were able to specifically target dopamine neurons in the VTA, and this light activated channel was able to travel down axons to the nucleus accumbens, and so we can stimulate dopamine transmission in the nucleus accumbens. And to further investigate astrocytes in the nucleus accumbens, we targeted a calcium indicator to astrocytes in the nucleus accumbens. And then we had a fiber optic channel to be able to record astrocyte activity. And this is a schematic of our preclinical model where we have an awake behaving animal connected to our fiber photometry system, which can both optogenetically stimulate dopamine transmission and also record calcium in astrocytes. And here's a video of our animal freely exploring its environment, hooked up to our system. And when we activate dopamine transmission indicated by these red markers, there's a robust increase in astrocyte calcium fluorescence, showing that astrocytes do indeed respond to dopamine transmission with increases in calcium. This increase in calcium was reproducible. We were able to stimulate dopamine transmission 5 times, and each time we did see this robust increase in astrocyte calcium activity, and our response to calcium in astrocytes was dose dependent. So with higher frequency of stimulation of dopamine transmission, we saw a higher increase in astrocyte calcium responsiveness to dopamine. Importantly, as we show here in control conditions in our histogram, there's an increase in astrocyte calcium levels. And when we block dopamine signaling with the global dopamine receptor antagonist flupentixol, we see that there's no longer an increase in calcium in astrocytes, showing that astrocytes are responding to dopamine through dopamine receptor activation. So far, what I've shown is that in awake behaving animals, astrocytes can respond to dopamine transmission with increases in calcium, suggesting that they're able to play an active role in dopamine signaling. So next, what we wanted to know is what are the cellular mechanisms of astrocyte responsiveness to dopamine? To do this, we transitioned to slice experiments where we had acute coronal slices from the mouse brain that contained the nucleus accumbens. And we're able to structurally localize our astrocytes using SR101, which is a structural marker for astrocytes. And again, we wanted to look at the calcium levels in astrocytes using a calcium indicator. And we're able to see that both our structural marker and our calcium indicator are co-localized in the nucleus accumbens. For reference, this is the anterior commissure. So when we were able to record calcium in astrocytes in slices, we did 2 different experiments. We either locally applied dopamine to the slices, or we optogenetically stimulated the dopaminergic transmission like we did in our awake behaving animals. And what we found was that astrocytes do indeed respond to dopamine in our brain slices as well. So I'm going to show a video of astrocyte responsiveness to dopamine in the nucleus accumbens. In this video, you will see a white circle that shows up here, indicating when I apply dopamine, and you'll see this robust response to calcium, to dopamine with increases in calcium. So these experiments I loved doing in Alfonso's lab. These are some of the joys in life when I was conducting my PhD work, seeing this robust response in calcium in astrocytes. And if we focus on one astrocyte here, for example, this astrocyte, you can see where it got its name of astrocyte, which is a star-like shaped cell. So this astrocyte definitely has a star-like shape with its cell body and its processes extending out from away from it. And so we're seeing that astrocytes do indeed respond to dopamine with increases in calcium. So next, now that we know that astrocytes respond to dopamine, we wanted to know what are the cellular mechanisms of this response. So I'll walk through this figure with everyone. So essentially, this is an astrocyte in baseline conditions. When we apply dopamine, there's a robust increase in the relative fluorescence in our astrocyte. And to further show that astrocytes respond to dopamine, we have our relative fluorescence traces. The arrow indicates when I apply dopamine, and we see this increase in calcium in our relative fluorescence in our astrocytes, also represented by a raster plot, where in baseline conditions, this is the amount of activity that's happening. At time point 0, we see I apply dopamine, and there's this robust increase in activity of our astrocytes. And in our lab, we really like to look at astrocyte calcium signaling with calcium event probability, which essentially is at any given moment, what is the likelihood that astrocytes are active? So in baseline conditions, about 20% of astrocytes are active. And then when we apply dopamine, there's a significant increase to about 50 to 60%, suggesting that astrocytes do indeed respond to dopamine with increases in calcium. We also represent this in our histogram plot. So essentially in control conditions, we see that there's a significant increase in calcium in astrocytes. And like our in vivo awake behaving animal experiments, this increase in calcium is ablated in the presence of flupentixol, which is our global dopamine receptor antagonist, suggesting that astrocytes are indeed responding to dopamine through dopamine receptor signaling. And importantly, we were able to find out that astrocytes are specifically responding through D1 receptors because in the presence of our D1 receptor antagonist, sharing 23390, we no longer see an increase in calcium in response to dopamine, but we still see an increase in calcium in response to dopamine in the presence of our D2 receptor antagonist sulfuride, suggesting that astrocytes are responding to dopamine through D1 receptors, because in the presence of our D1 receptor antagonist, we ablate that response, but it's still present in the presence of our D2 receptor antagonist. So we're able to show that astrocytes respond to exogenous dopamine via D1 receptors. We were able to recapitulate our results looking at endogenous dopamine signaling with synaptically released dopamine using our optogenetic experimental model. And essentially, when we activate synaptic dopamine release, we see that there is still a robust increase in cytoplasmic calcium in astrocytes, also represented by our relative fluorescence traces and in our raster plot. And again, when we look at the pharmacology, astrocytes in control conditions respond to dopamine and have this significant increase in calcium. In the presence of our global dopamine receptor antagonist, we no longer see an increase in calcium in astrocytes, and we no longer see an increase in calcium in response to our D1 receptor antagonist, suggesting that astrocytes are responding to synaptically released dopamine via D1 receptors. So far, what I've shown is that dopamine increases calcium in astrocytes, and this is through D1 receptors. So next, what we wanted to know is whether or not astrocytes express D1 receptors. To do this, we collaborated with Dr. Lujan, who conducted electron microscopy experiments in the nucleus accumbens. And what he found was that indeed, astrocytes in the nucleus accumbens do express D1 receptors. So they contain the machinery to respond to dopamine via D1 receptors. So now that we know that astrocytes respond to dopamine with increases in calcium, what we wanted to know is how does this influence the neurons? We want to show that astrocytes are an active player in synaptic transmission and plasticity. So what is going on with the neurons when this increase in calcium happens? So essentially, our next part of the experiments is assessing whether or not astrocytes contribute to dopamine neuromodulation. So before my experiments, it was already known in the literature that dopamine depresses excitatory transmission. This was already known in the literature. So what we wanted to know is how do astrocytes contribute to that dopamine depression of excitatory transmission? To do these experiments, we recorded from neurons in our slice prep and electrically stimulated excitatory transmission while applying dopamine. And what we can see is that when we record from neurons, we're able to record this excitatory postsynaptic current. And in the presence of dopamine, this excitatory current is depressed. This was already known in the literature. What we're bringing to the table is astrocytes. So as shown before, when we apply dopamine, there's a robust increase in calcium in astrocytes, and there's a depression in excitatory transmission. And interestingly, these two phenomena are significantly correlated. So dopamine both depresses excitatory transmission and increases calcium in astrocytes, and these two events are correlated. This was also seen when we looked at synaptically released dopamine using our optogenetic approach. So essentially, we're still able to record excitatory postsynaptic currents in neurons. When we optogenetically stimulate dopaminergic transmission, we see a depression in excitatory postsynaptic currents, and we also see an increase in calcium in astrocytes. And these two events are significantly correlated. So far, what we've seen is that dopamine can both increase calcium in astrocytes and depress excitatory transmission in neurons. And these two events are correlated. But of course, as scientists, what we really want to know is whether or not astrocyte calcium is necessary for dopamine-evoked depression of excitatory transmission. So to do this, it's very important for us to understand how calcium signaling in astrocytes work. So when dopamine binds to the dopamine receptor, there are downstream signaling events that leads to the activation of an IP3 receptor to lead to the increase in calcium in astrocytes. To understand whether or not astrocyte calcium is necessary for dopamine-evoked neuromodulation, we wanted to block astrocyte calcium, taking two approaches. The first approach is to knock out the IP3 receptor type 2 in astrocytes to block that increase in calcium in astrocytes. And then the second approach was to knock out the dopamine receptor in astrocytes to see whether or not that activation was necessary for the effects on neurons. So when we look at our first approach, where we knock out the IP3 receptor type 2 in astrocytes, we see that there's no longer an increase in calcium in response to dopamine, and there's no longer depression in excitatory transmission, suggesting that astrocyte calcium is indeed necessary for dopamine-evoked depression of excitatory transmission. To further confirm these results, we took our second approach, which was to knock out the D1 receptor in astrocytes. So we used a transgenic mouse model that had the D1 receptor floxed, and then we injected a GFAP mCherry-Cree viral vector into the nucleus accumbens to specifically knock out D1 receptors in astrocytes in the nucleus accumbens. And what we found was that in our mice that have the D1 receptors knocked out, there again is no longer an increase in calcium in response to dopamine compared to our wild-type mice. Our wild-type mice have this beautiful increase in calcium in response to dopamine, but when we knock out the D1 receptor, we no longer see that increase in calcium in response to dopamine, and we no longer see a depression in excitatory transmission, suggesting that astrocyte calcium is indeed necessary for dopamine-evoked neuromodulation. So far, what I've shown is that dopamine acts on D1 receptors to increase calcium in astrocytes, and this increase in calcium in astrocytes is necessary for the depression of excitatory transmission in the nucleus accumbens. So next, what we wanted to know is, what are astrocytes releasing to cause this decrease in calcium, to cause this decrease in excitatory transmission in neurons? So again, in the literature, before our experiments were done, we knew that dopamine depresses excitatory transmission through D1 receptors, and this was through adenosine signaling. But nobody knew where the adenosine was coming from. And my project proposes that the adenosine is coming from the astrocyte. To do these experiments, we again recorded from neurons in the nucleus accumbens, stimulated excitatory transmission, and then applied dopamine. So the change in these experiments was to do them in the presence of the A1 receptor antagonist, CPT. So essentially, we're able to record our excitatory postsynaptic currents. And in the presence of our A1 receptor antagonist, we no longer see a depression in excitatory transmission, suggesting that A1 signaling is mediating dopamine-evoked depression of excitatory transmission. Importantly, this A1 signaling is downstream of calcium events in astrocytes, because even in the presence of our A1 receptor antagonist, we still see an increase in calcium in astrocytes, suggesting that astrocytes are responding to dopamine with increases in calcium. Then there is A1 signaling on board to cause the depression of excitatory transmission. So overall, what I've shown so far is that dopamine acts on D1 receptors in astrocytes to cause this increase in calcium. Astrocytes are then releasing ATP adenosine to cause a depression in excitatory transmission. So next, what we wanted to know, now that we've shown that astrocyte calcium is necessary for the depression of excitatory transmission, is astrocyte calcium also sufficient to modulate synaptic transmission in the nucleus accumbens? To do these experiments, we took out dopamine completely, and we just looked at what happens when we activate astrocytes. Does that modulate neuronal activity in the nucleus accumbens? To do these experiments, we targeted designer receptors to astrocytes in the nucleus accumbens, and these designer receptors are really cool because they are only activated by a designer drug, CNO. So essentially, once we're able to target astrocytes in the nucleus accumbens with our designer receptor, when we activate the astrocytes with our designer drug, we see this increase in calcium in astrocytes in response to CNO. And what's really interesting is that this increase in calcium in astrocytes co-occurs with a depression in excitatory transmission in the nucleus accumbens, suggesting that astrocyte calcium activity is sufficient to modulate synaptic transmission in the nucleus accumbens. So overall, in terms of an interim summary, we've seen that dopamine can act on D1 receptors in astrocytes to increase calcium. Astrocytes are playing an active role in dopamine neuromodulation by releasing ATP adenosine to depress excitatory transmission in the nucleus accumbens. And we've shown that astrocyte calcium is both necessary and sufficient for the modulation of excitatory transmission in the nucleus accumbens. So next, what we wanted to know is whether or not astrocytes also play a role in amphetamine neuromodulation. To do these experiments, we took advantage of the fact that amphetamine modulates dopamine levels at the synapse. So in baseline conditions, dopamine is released at the synapse, and to maintain physiologic levels of dopamine, it is transported out of the synapse through a dopamine transporter. But with amphetamine on board, amphetamine both blocks and reverses the dopamine transporter to lead to a robust increase in dopamine levels at the synapse. And what we wanted to investigate is whether or not astrocytes respond to this increase in dopamine at the synapse. Does amphetamine modulate astrocyte responsiveness to dopamine? So we went back to our preclinical awake behaving mouse model, where we were able to record astrocyte activity in the nucleus accumbens in response to dopamine. So the red bar indicates when I synaptically stimulate dopamine release. In basal conditions, there's a robust increase in calcium in response to dopamine. And with amphetamine on board, there's a significant increase in astrocyte responsiveness to dopamine, suggesting that amphetamine is modulating astrocyte activity in awake behaving animals. And this is reflected both in our relative fluorescence and the rise time of the calcium levels, and also in the response width of the calcium response in astrocytes. So astrocytes respond to dopamine, and this responsiveness to dopamine is modulated by amphetamine. So next, we went back to our SLICE experiments, where we looked at astrocyte activity in basal levels. These are relative fluorescence traces. And in the presence of amphetamine, there's a significant increase in astrocyte calcium activity, also represented by a raster plot. And importantly, this increase in calcium in astrocytes is also co-occurs with the depression in excitatory transmission in neurons, suggesting that astrocyte calcium activity and amphetamine-evoked depression of excitatory transmission are correlated. And to show that astrocytes actively participate in amphetamine modulation of neuromodulation, we again showed that astrocyte calcium is necessary for amphetamine-evoked depression of excitatory transmission using our IP3 receptor 2 type knockout model. So essentially, in our mice that have the IP3 receptor 2 knocked out, we no longer see an increase in calcium in astrocytes in response to amphetamine, and we no longer see a depression in excitatory transmission. And in our GFAP D1 knockout animals, where we knock out the D1 receptor in astrocytes, we no longer see an increase in calcium in astrocytes, and we no longer see a depression in excitatory transmission, suggesting that astrocyte calcium is also necessary for amphetamine-evoked neuromodulation. So now that we know that astrocytes contribute to amphetamine neuromodulation, we also wanted to know whether or not astrocytes play an active role in the behavioral effects of amphetamine. So one really interesting thing about amphetamine is that when animals are exposed to amphetamine, they have a robust increase in motor activity and psychomotor activity. So essentially, if we look at this plot of a wild type mouse in baseline conditions, they received saline, they're exploring their environment, and then when they have amphetamine on board, there's a robust increase in their locomotor activity. And what we found was that when we compare this increase in locomotor activity in our wild type animals compared to our animals that have decreased calcium signaling in astrocytes, there is an attenuation in the psychostimulant effects of amphetamine, both in our IP3 receptor type 2 knockout animals and in our animals that have the D1 receptor knocked out in the astrocytes, suggesting that astrocyte calcium activity also contributes to the psychostimulant effects of amphetamine. So overall, in this presentation, I've shown that astrocytes respond to dopamine with increases in calcium through D1 receptor signaling. This leads to the release of ATP adenosine signaling to activate A1 receptors to depress excitatory transmission in neurons. And astrocytes play an active role in both dopamine neuromodulation and amphetamine neuromodulation. And overall, I would like to propose that astrocytes modulate dopamine signaling and brain reward signaling, and could serve as novel therapeutic targets for diseases with disrupted dopamine signaling, such as drug addiction. And with that, I would like to thank both the Racke Lab, my mentor during my PhD work, and then also my co-mentor, Dr. Mark Thomas in the Thomas Lab. Of course, my thesis committee. I'd also like to thank the Rothwell Lab, where I did all of my photometry experiments in, and then the core facilities at the University of Minnesota. If anybody's ever doing any type of experiments at Minnesota and wants to have more expert opinions, please contact the core facilities. They are amazing. And of course, I would like to thank my funding sources as well. And with that, I will take any questions.
Thank you, Michelle, for the wonderful talk. I want to say that you explained it very clearly, and the experiments look very easy. They are not easy, and it requires a lot of work. I want to clarify that for the audience. And from that, let's say go for the first question. No questions? I'm going to start with the general questions I want to ask you. I want to know your opinion. Do you think this could be assets will be involved also in other, you're showing here the VTA, nucleus accumbens, dopaminergic signaling. Another dopaminergic signaling with a very important function is the projections to the cortex, the frontal cortex.
Yeah.
Do you think that would be similar mechanism? What were you thinking about your thoughts about that? Would be the same or not?
So essentially, there has been experiments conducted in other labs showing that astrocytes do indeed express dopamine receptors in the prefrontal cortex. So with that machinery in place, astrocytes could respond to dopamine with increases in calcium. But I do think that astrocytes modulate synaptic transmission based on the environment that they're in. So in terms of the specific mechanisms that are mediating this excitatory transmission, it could be different in the prefrontal cortex. I haven't conducted the experiments, but it could be that astrocytes modulate excitatory transmission, but maybe not inhibitory transmission. And then it'd also be really interesting to investigate based on the input to the circuit, how do astrocytes modulate that transmission as well.
I have a question here. Can you speculate to the impact that this research may have to understanding pathophysiology of psychiatric disorders?
Yes, absolutely. So the amazing thing about the brain and psychiatric disorders is that there are multiple circuits and inputs that are contributing to the physiology and the phenotype that we see in patients with psychiatric disorders. So essentially, when we are thinking about how circuits are disrupted in brain disorders and psychiatric disorders, it's really important to understand all of the components that are contributing to that signaling mechanism, in addition to the neurons and the astrocytes. And one really interesting thing showing that astrocytes express dopamine receptors and respond to dopamine signaling, when we are thinking about designing new therapeutics to target dopamine signaling, it might be interesting to specifically target astrocyte dopamine receptors versus neuronal dopamine receptors. So that's one way to kind of apply this knowledge to new advances in treatments for psychiatric disorders. I also want to point out, I love non-invasive neuromodulation. I think it's amazing and it's super exciting. And there is work looking at the role of astrocytes in non-invasive neuromodulation. And so a group looked at direct current stimulation. And what they found was that astrocyte calcium was contributing to the antidepressant effects of direct current stimulation in a mouse model of depression. So it could be that all of these therapeutics and interventions that we're using right now, where we don't know what the mechanism of action is, it could be going through the astrocyte. So it's so important for us to understand what are the circuits that are implicated in disease processes, what are the cell types implicated in disease processes, and how can we make very specific therapeutic targets to minimize the side effects that we see in our patients.
Another question, if you propose astrocyte modulation as a target for disorders of reward signaling, how can we develop therapeutic candidates specific only to astrocytes?
Yes, thank you. Thank you for the question. So I did just mention the direct current stimulation, which I think is probably acting through astrocytes. And the other really interesting thing about non-invasive neuromodulation in general is that we can set the parameters. So whether it's direct current stimulation or transcranial magnetic stimulation, we are setting the parameters for the frequency, the duration of the stimulus train as well. And so it could be that at some frequency stimulations, at some parameters, we're targeting neurons, and at other parameters, we're targeting astrocytes. And I think that'd be a really fun experiment to conduct to really understand how can we target specific circuits and specific cell types. And then another avenue to specifically target astrocytes would be through adeno-associated viral vectors. So there are a lot of AAVs right now being investigated for the treatment of brain cancers, so essentially targeting AAV viral vectors for specific treatments of brain cancer. And it would be really interesting to see whether or not we can also use AAV viral vectors for targeting of specific cell types, including astrocytes. Throughout my work that I showed in this presentation, I was able to specifically target astrocytes using AAV viral vectors to specific brain regions. And so since we're right now looking at that use in the human population, I think moving forward, we can also be able to use the technology to target specific cell types, specific receptors as well.
What do you think this might mean for dopamine deficit disorder, especially Parkinson's? How might this mechanism be druggable or otherwise targeted?
Yeah, thank you for the question. So before I even joined Alfonso's lab, he did some experiments in the striatum looking at astrocyte regulation of synaptic transmission in the striatum. And what he found, what the project found was that essentially there are D1 network astrocytes and D2 network astrocytes that can modulate specific circuits and specific synaptic transmission as well. And so in terms of looking at the findings for example, Parkinson's disease, is when we are looking at this dopamine deficit, is there also a deficit in astrocyte responsiveness to dopamine? Because if there is, let's say there's a decrease in dopamine, but then there's also a deficit in the astrocyte responsiveness to dopamine, maybe we can activate astrocytes. If astrocytes are modulating the phenotype that we're seeing in our patients, what if we can activate astrocytes without the dopamine on board? I showed those experiments in this presentation as well, where we were able to use a designer receptor using our adeno-associated virus, and we're able to activate astrocytes, and we're able to recapitulate the effects of dopamine on those neurons. So maybe we can activate astrocytes without dopamine on board and really be able to help our patients as well. So those would be really fun experiments to do.
It is an avenue to target ADHD, schizophrenia, and addiction. I think you have answered that. Do you want to comment more on that?
Can you please repeat the question?
Do you see this as an avenue to target ADHD, schizophrenia, and addiction?
Absolutely, yes, absolutely. And the really cool thing about like ADHD is that the treatment would be amphetamine, and I just showed that amphetamine modulates astrocyte activity. So I definitely think that we can use the astrocyte involvement in psychostimulants and dopamine signaling to be able to treat disorders.
Another question. I'm wondering if you have any thoughts how astrocytes and dopamine may fit into the dopamine hypothesis of schizophrenia. Most antipsychotics are D2 antagonists. Is there a role for astrocytes in this process?
Yeah, absolutely. So I love this question because it also shows that for disease processes and also astrocytes, they have different characteristics based on the brain region. So in the nucleus accumbens core, which I looked at, I saw that astrocytes respond to dopamine via D1 receptors. There were some experiments in another lab that looked at astrocyte responsiveness to dopamine in the hippocampus. And they saw that D1 receptor activation leads to increase in calcium in astrocytes, and D2 activation led to a decrease in calcium in astrocytes. So it'd be really interesting to really understand what circuits specifically do we want to modulate in schizophrenia, and then how are astrocytes involved in those specific circuits? Is it through D1 receptor signaling? Is it through D2 receptor signaling? And then how can we like specifically target astrocytes to modulate dopamine signaling to help our patients have a better quality of life moving forward?
Any more questions? I've received several comments, many comments, congratulating for the great presentation. I fully agree with them.
Thank you.
Any other question? No? Okay. Thank you, Michelle, for the great talk. It's been really great. So I want to thank you all for your participation, and thank you, Michelle, for the great presentation.
Yes. Thank you, everyone, so much. It was really fun being able to present my work to everyone. And if you have any other specific questions, please feel free to email me. Thank you.
Okay, bye-bye, everybody.